So today we are going to focus on Protozoa. So we'll start by looking at the Protozoa handout. So this is the handout, and it's one that I actually made last spring when we went remote, just to sort of simplify and show you what we need to do. And I have a bunch of videos on Protozoa that we'll watch. But we're going to talk about parasitic protozoa as well as free-living protozoa. But we'll start with the parasites first. We have two parasites we're going to talk about. One is Plasmodium, and the other is Trypanosoma. So Plasmodium is a protozoan parasite that causes malaria, and it is transmitted by mosquitoes, but only female mosquitoes, not males. Only the female mosquitoes bite and suck blood, but not the males. So why is it only females are bloodsuckers? So why only females bite?
Is it for their larvae or something?
Well, not larva, right? Not in this case. We don't use the word larva, but for their eggs, right? For the egg development. So in order to help their eggs to develop, they need the extra nutrition, right? And the males don't need that because what do males contribute? Males are just sperm donors, right? So they just contribute chromosomes. Everything else is in the egg from the female. So she needs the extra nutrition, right, to help her eggs to form and develop. So only the females bite. And unfortunately, because mosquitoes do bite, they can transmit diseases like malaria. Now, in the United States, we don't have a problem with malaria anymore. A long time ago we did, but through management of wetlands and other things, we eliminated malaria. But that does not mean that malaria cases still do not happen. In fact, there were 2 cases that happened in our area. I forget, it's like 10, 15 years ago. And what happened is they were on one of the islands in the Potomac River, and they got bitten by mosquitoes and they got malaria. And so when this was reported, to health authorities, they were like, well, this is really unusual. How could you get malaria here? And so they went to investigate, and they set traps on the island and captured mosquitoes and tested them. And sure enough, they found some were infected with malaria. So how did malaria get here? Well, people do travel into and out of the country, and they go to other countries where malaria is a problem. And so it's possible for someone who's infected with malaria to get bitten by a mosquito, and the mosquito picks up the malaria parasite, and that's how the chain of transmission can start. So it is possible, but very unlikely, to get malaria here. But there are other diseases, in particular viral diseases that we'll start talking about next week, which are transmitted by mosquitoes. So it is important to protect yourself against mosquitoes. So what can you do to protect yourself? What can you do to protect yourselves from mosquitoes? Doesn't anyone get bitten by mosquitoes? What do you do to protect yourself?
We wear long
Oh, we put it.
so it goes your skin.
Right, so you could wear long sleeves, long pants. What else?
Put off spray.
Right, so you can use an insect repellent like DEET or picaridin, which, help to stop mosquitoes from biting you. You can avoid being outside during dusk and dawn, when the sun's coming up or when the sun's going down, when mosquitoes are most active. If you have any standing water on your property, you should turn those things over and empty the water because mosquitoes will use it to breed. So there are things you can do to minimize being bitten, but it is hard to completely avoid being bitten by mosquitoes because there's a lot of them in our area and they do like to bite. And some people are more attractive to mosquitoes than others, right? And when it comes to blood type, mosquitoes do have a preference, and they prefer type O blood. I don't know why they don't like AB. Maybe they don't like the taste of the antigens on the surface of the red blood cells, but they do prefer type O blood. And so some people are just much more attractive to mosquitoes than others. And I'm one of those people. Mosquitoes absolutely love me. I mean, they will, fly right by other people and come straight to me and bite me and not anyone else. Because that actually happened once. I was in a classroom teaching, and the door was on the other side of where I was. And this mosquito flew in, and I could see it because it flew in front of the projector, so you could see the mosquito. It was lit up. It flew by all these students sitting in the front row and came straight over to me. It had a lot of options, but it came straight for me. So some people, mosquitoes just like a lot better than others, right? They smell better, they taste better, and so mosquitoes love them. Now, most people do get a minor allergic reaction when they get bitten, right? We're allergic to the antigens in their saliva, so we get a little red bump that's itchy. And there are some people that actually don't have any reaction. And some of those people tend to say, oh, well, I never get bitten. No, they probably do get bitten. It's just that they don't react, so they don't know they've been bitten. So not everyone reacts. Most people just get a small itchy bump. It may itch for 15 minutes or half an hour or something. And then some people actually get a very large reaction. I had a student once had a very large red area, like 6 to 8 inches on her shoulder, where she'd been bitten by a mosquito. She was just extremely sensitive to them. So some people are very sensitive, others are not. Some people are very attracted to mosquitoes, others not so much. But it is important to try to avoid being bitten, but that's really hard to do because if you have a yard or you like to be outside and do things, mosquitoes are always around. Our native mosquitoes are not as much of a problem because our native mosquitoes, they hate the sun. So that's why they like dusk and dawn, or if it's a cloudy day, or if you're under trees or surrounded by a lot of dense foliage, you'll have mosquitoes in there. And our native mosquitoes, this is what they do. They very gently land on you. And I'm talking about the ones around here. They land on you, they gently pierce your skin, suck your blood, and then they fly away, right? So the mosquito takes her fill and she flies away. And you may never even know that you were bitten until it starts to itch. The problem is that, and our native mosquitoes are slow, so they're easy to spot. We have invasive mosquitoes here, in particular the Asian tiger mosquito, which we have in Maryland, in Montgomery County, because I've had them on my property because they're striped, and that's how you can tell they're Asian tiger mosquitoes. And they're very different. One, the sun does not bother them. They will bite you in full sun. They are more aggressive in the sense that they land on you, they bite you, and she sucks a little bit of blood and then she takes off. And then she comes right back and bites you again and sucks a little bit more blood and takes off. And then comes back and bites you again and sucks some more blood. So an Asian tiger mosquito may bite you 3, 4, 5 times before they get all the blood they want. Our native mosquitoes just bite you once, suck the blood, and if you see them, they're so slow, you can easily spot them and kill them. Asian tiger mosquitoes, they fly around a lot more, so they're harder to kill. And the Asian tiger mosquitoes need very little water to reproduce in. If you just have something like a bottle cap from a water bottle, that's enough water for them to lay eggs and develop in a little bottle cap of water. So Asian tiger mosquitoes are more aggressive. They bite you more often. They bite in full sun and need very little water to reproduce in. So those are much more of a problem than our native mosquitoes. So it's important to try and protect yourself, but even repellents are not 100%. Although DEET is the best one, it's been around for almost 100 years, it's very reliable, it's relatively safe. Nothing works better than DEET, although they have alternatives which may not smell, like picaridin doesn't smell once it's dry, or they have what I think is a nasty smelling lemon eucalyptus mixture, which I think smells worse than DEET. But there are other things, but nothing is more effective than DEET. But even DEET, they've done studies where there are mosquitoes, they come up to your skin, they're like, nope, right, because you've got DEET on, and they fly away. But maybe if you sit there for a couple of hours, that same mosquito comes back and says, you know what, I need blood, and they bite you anyway. So they have shown that some mosquitoes, they just decide they don't care anymore about the DEET, right? They're repelled initially, but they want that blood meal so badly that they come back and bite you anyway, even if you're wearing DEET. So some repellents are not as effective as they used to be because the mosquitoes are just deciding they'd rather take the blood. Even if your skin is repellent to them, they still want that blood. So it's getting harder and harder, to deal with mosquitoes. And as non-existent global warming keeps occurring, our mosquito populations and problems are going to increase. And all these diseases that are found in the southern hemisphere are going to migrate northward as the northern hemisphere gets warmer and warmer. So it's important to try to avoid being bitten. It's almost impossible if you like to be outdoors, but do your best to try to avoid being bitten. But back to Plasmodium. So it's transmitted by mosquitoes. It causes malaria. And it is an intracellular parasite, so it lives inside of red blood cells. And like most protozoa, Plasmodium has a complex life cycle with many different stages, but we're just concerned with one stage, and that's the signet ring stage. And we can see in these images, we have these rings inside of red blood cells. And so this is Plasmodium. When you actually look at a blood that's infected with Plasmodium, you'll find all the different stages there, but we're just concerned about signet ring stage because it does look like a ring, and depending on the type of staining that's done, it often looks like a ruby ring with a red dot and kind of a purple circular body. You can have more than one signet ring inside of a cell, and here we see twins, right? I've even seen images of triplets where there's 3 signet rings inside of a single red blood cell. So, but we do have drugs to help treat malaria. Malaria is endemic in many countries and many continents like Africa and Asia. And so an interesting evolutionary adaptation has occurred to deal with malaria. What is that adaptation?
Is it sickle cells?
Yep, sickle cell disease, right? So you probably have heard of sickle cell disease or sickle cell anemia. It's where the hemoglobin is abnormal and it changes the shape of the red blood cells, and they actually turn to like a quarter moon shape. They're curved, or like a sickle, the blade that's used to slice through wheat and grasses and things. So, and the problem is, one, the hemoglobin doesn't carry oxygen as efficiently anymore, and the abnormal shape of the red blood cells, they get stuck in capillaries, and that can lead to organ damage and organ failure. Now, when it comes to genetics, remember we have two copies of a particular gene. Would it be called different versions or different copies of the same gene? What do we call that?
Alleles?
Alleles, right? Alleles. Remember, you have two alleles for every gene, one from mom, one from dad. And so if you have the mutant allele, right, the sickle cell allele, if you have just one of them and the other allele is normal, you are heterozygous. And you also have resistance to malaria, right? Because the parasite doesn't like the abnormal hemoglobin, so it can't reproduce inside of the red blood cells. So that's good. If you got one good allele, one bad allele, then you're phenotypically normal, you're healthy, and you have some resistance to malaria, so that protects you from malaria. The problem is when you're homozygous for the sickle's allele. If you have two copies of the mutated allele for hemoglobin, then you have actual sickle cell disease. And before modern medicine, that was a lethal characteristic. So most children born with sickle cell did not grow to become adults. They would die. So then you were left with your population, which if you do the Punnett square, you know that you have 25% are normal, right? Two normal copies of the allele, and 50% are heterozygous, and then 25% were homozygous, but they would die out. And that would leave you with 2/3 of the population protected from malaria, right? Because 1/4 died from the sickle cell trait. And since you had so many people that were resistant, you know, the mosquitoes, the chances were the mosquitoes would bite the resistant people and not the ones who are susceptible. And so that way they gain protection. So that was the benefit of the sickle cell trait. Having one allele gave you resistance to malaria and helped to protect you, because malaria was also a serious disease with a high mortality rate prior to modern medicine. And so nature found a way to deal with that. And so the sickle cell trait evolved in Africa, it evolved in Asia as a way to protect people from malaria. But there was a price to pay. And that price was that about 25% of your population would die from sickle cell disease. But the remainder would be protected, right, from malaria. Of course, today, like I said, we have drugs to help protect against malaria. So malaria is not as big a deal now. And so the sickle cell trait has become more of a negative thing rather than a positive thing. I had a student once in micro, and this was a long time ago, and she had sickle cell disease. And she was in her 40s, so she, you know, with the help of modern medicine, was able to survive. But she wanted to actually look at her blood. And since this was a long time ago, I was still able to, you know, prick her finger and take a blood sample. And so we all got to see her blood, and she had sickle cells in her bloodstream. So she clearly had sickle cell disease, but most of her red blood cells were still normal. But periodically she would have episodes where they, you know, have a lot more sickle cells. So, but it was really interesting to be able to see actual sickle cells from a living person. So nature often helps to find a way to protect, but there may be some consequences. There may be a price to pay for that protection. So any questions on Plasmodium and malaria? Our next parasite is Trypanosoma. And so this causes sleeping sickness, which is what we call the disease in Africa, but it also occurs in South America, but there they call it trypanosomiasis instead. And this is transmitted by a fly. Now, depending on where you come from, how you pronounce this is different. Here in the States, we call it the tsetse fly, but in other countries, you may call it tsetse or cheche. But whatever, it's a fly that we don't have here, so we don't worry about trypanosoma in the United States. But it is a significant parasite. It's an extracellular parasite, so it's large. It is in the bloodstream, but it's outside of cells. And it is flagellated, so you can see they have a flagellum. So it's a large parasite. It's easy to see in the blood. Now, what you need to remember about the parasites, you need to know what disease they cause, how they're transmitted, and whether they're intracellular or extracellular. And this is very important because on the lab exam, you're going to see these. And if you mix them up, you're going to miss a lot of questions and a lot of points. So it's important to know the difference between the two. What they are, what disease, what transmits them, if they're intracellular or extracellular. So any questions about these two parasites? So when it comes to the free-living protozoa, I'm just going to talk and show you a bunch of videos. But the main idea here, you guys would spend a lot of time in lab making wet mounts. We put a drop of your organism onto a slide and then put a cover slip on top, and then go and try and find these things, which was easy to do because they're much, much larger than bacteria. So much more fun to look at these because they're big. And then try and figure out how are they moving. And with the protozoa, they can use flagella or cilia or pseudopods. Remember, bacteria only use which one?
Flagella.
Right, flagella. So I do often like to ask a question and be kind of tricky and use the words protozoa and prokaryotes interchangeably. And so if I say, what structures do protozoa move? It's all of them. Used to move is all of them. But if I ask what do prokaryotes use, it's just one of them, right? So I try to make you look carefully at the questions and pay attention to the detail. Now, when you make your wet mounts, just like we talked about with the bacterial wet mount slides to determine motility, you have to close the iris when you start because you're just focusing through water. And so you need to dim the light so you can find something. And usually you'll see something moving around, and then you try and zoom in on it by increasing the magnification. Now, some of these things, like paramecia, they move fast. So you can't really keep them in your field of view. So what we would do is mix a thick solution called methyl cellulose. The brand name is Protozoa, but it's just methyl cellulose, which is thick, kind of like syrup. And you mix it with your sample, and that slows down these fast protozoa, so you can actually sit and focus on them. Other things like the amoeba, they're so slow that if you use protozoa, you wouldn't see anything with them. If you use protozoa, you wouldn't see anything with them. But we would often mix some protozoa, right, the methyl cellulose, with some of the faster moving ones, like paramecia, sometimes with euglena. But euglena really don't like it. They don't react well to it. So let's look at some videos. And I got a bunch. So we can look at the amoeba first. And so this is made with a phase contrast microscope, which is why the amoeba is all kind of shiny and glittery. And we see pseudopods being extended in the amoeba. So the amoeba, projects its plasma membrane in one direction and then shifts all the cytoplasm, including all the organelles, in that direction. And all these little things you see inside, those are all the organelles inside of that amoeba. But they do move slowly. They crawl around on the bottom of your culture dish in lab or at the bottom of ponds and lakes and things like that. And they wander around looking for things to eat. And then in a few minutes, I'm going to show you some videos that I made of amoeba eating some little protozoa. But you can see the pseudopods extending, and so the amoeba just kind of crawl around. When it comes to euglena, here we have euglena, again a phase contrast microscope. And so what structure can you see?
Flagella.
Right, so they do have one flagellum on the end, and they are in this thick solution, this methyl cellulose, which is why they're not swimming around, because normally they would be very, very active and swimming around, but they've been slowed down. Now, you notice that they have something green inside them, right? They have chloroplasts inside them. So what does that mean they can do?
Can they do photosynthesis?
Yep, they can photosynthesize, right? So like a plant. But they swim around like an animal, and they can also eat food like an animal. So euglena have characteristics of more than one kingdom. That's why they're put into the kingdom Protista, right? Because they have characteristics of more than one kingdom. They're photosynthetic, but they swim and they eat food. Here we have a video on different kinds of
Many
material.
cells use flagella for moving around. These Chlamydomonas swim very fast using their two flagella. At high magnification, the two flagella become visible when a cell is embedded in mucilage, slowing their activity right down. Like many algae, Chlamydomonas has an eye spot which responds to light and affects the way the flagella beat, steering the cell toward or away from it. Many protists have flagella of different length and function. This euglenoid, called Phacus, is swimming in the direction of its longer flagellum. These relatives of Phacus also have two flagella which function differently. One beats in a coil around the cell. The other, as in Phacus, extends forwards. Other algae have more than two flagella. This cell has 8, inserted into a deep pit. It normally swims very rapidly and had to be slowed down to have its picture taken. Large protists like Paramecium swim rapidly using hundreds or even thousands of flagella, or cilia as they are usually called. They are very agile as they search amongst the pond debris for food. Beating of the cilia is coordinated, and the direction of beating is reversed when the cell changes the direction it wants to go. This cell, like the last ones, had to be slowed down in a sticky solution so that it could be photographed. Many protists also use their cilia to create currents for feeding, carrying nearby organisms close enough to be eaten. These algae are too big for this protist, but show the currents clearly. This large protistan cell called stentor feeds like a vacuum cleaner. A circular array of cilia around the flattened end of the cell sucks nearby organisms into a conical mouth where it can check out what it has captured. It is feeding here on green algae. Notice that it is quite selective about what it eats, swallowing some of the captured cells and rejecting others. The cells of some tissues in animals have multiple cilia, like these cells cultured from lung tissue. The activity of the cilia is coordinated so that liquid or mucilage can be moved long distances over the surface of the tissue. Flagella and cilia contain a core of microtubules. Using polarized light for imaging the cells, the microtubules show up as a dark or light fiber inside each cilium.
Remember, YouTube doesn't translate well, so the closed captioning wasn't very good. One of the organism is not a faker. It's not fake. It was facus, Phacus, but YouTube doesn't know that word. And the guy was British, so he says algae instead of algae. Either way is fine. But we've seen how a protozoa can move or use their cilia for feeding. So I have some videos that I made. And so if you go to my web page, you'll see that I have videos here. But when it comes to the videos, if you're not using Internet Explorer, this is what you get, all this stuff here. If you just highlight the first part up to the end of the number and you paste that, then it takes you to the podcast site. So here's the podcast site, right? So there is no podcast site. If you ever go here, almost everything here is mine because nobody uses the MC podcast site. So almost everything is mine. So if you do a search, you can find things on here that are from me. So here, just a healthy amoeba. And I made these in lab using the phase contrast microscope that we have in lab. And so here we just see an amoeba just doing its normal thing. And we also see these little cells, these little rods, which are actually bacteria that are in the water with the amoeba. Notice all the organelles are very shiny and shimmery because of the phase contrast. It's extending a pseudopod over here, shifting everything in that direction. And then we have a little protozoan swimming around there. It's much smaller than a paramecium is. So I also took some video of an amoeba eating. So what we have is a dumb little protozoan here who's bumped into the amoeba, and the amoeba is going to sense that. And eventually what the amoeba will do is extend its pseudopods around it to engulf it. And it takes a while for that to happen, so I'm just going to jump ahead here. So here you see like the pseudopods moving out to surround that poor little protozoan. And eventually it's going to completely surround that protozoan and engulf it. So now that protozoan is trapped.
How come it didn't leave before?
Well, it doesn't have much of a brain. Usually what happens is once these little things get surrounded, then they start to try and move and get away, but then it's too late. and they're trapped. And the amoeba can even engulf something large like a paramecium. I was just playing with the light when I was filming this. Let's see. And now it's realizing it's stuck and trying to get away, but it can't. So it's going to be engulfed and digested. So, but it's still alive. But it's gone. I made another video, again, of an amoeba eating. And here it swallowed this one already, or surrounded it. And then it's going to go after another one, or we'll see another amoeba going after another one. But here's one of these little protozoans. It got surrounded and trapped. So it tries to escape, but it's too late. So let's move this ahead. So here we have another one of these guys, and they're not very bright. Bumps up against the amoeba, and the amoeba's like, oh, look, something else I can eat. And this amoeba is very quickly going after it, so it must be really hungry. So it's trapped now, it's surrounded. It's going to be engulfed and digested. But at least these little protozoa have the satisfaction of knowing that the amoeba are going to die soon too. Because when we do this lab and you're finished with these slides, because they're wet slides with live organisms on it, standard procedure is to put the slides in a dish of disinfectant. So that's going to kill everything. But as we're going to see, amoeba will eventually explode because you put a cover slip on top of the drop. And remember, a cover slip is just a little thin piece of glass you put on top, but the cover slip is slowly sinking, and it will squish the amoeba and cause it to explode. So I do have videos of amoeba death, which I'll show you. So this one, let's see. Oops. Yeah, so amoeba death. So now you can tell the amoeba are getting close to dying because they get round, right? Because that cover slip is on top of them, pushing down on them, and so it makes them round. And we have lots of these little protozoa in here. And we also have some nice little motile bacteria that you'll see all throughout this video. These little black rods are motile bacteria. So, and of course we have this large structure inside. What's the big round thing inside? What's the big round structure inside? What is it?
Is it a vacuole?
Nope. What else is usually big inside of a cell? A eukaryotic cell. What? It contains the chromosomes. So what is it?
The nucleus?
Nucleus?
The nucleus, right? So I'm going to advance this. So we're getting close. The amoeba's getting misshapen. You can really see the membrane. It's got the halo around it where the membrane is. And you're going to see the membrane is going to pop. It's going to explode. And all of its guts are going to leak out. Remember, the guts for an amoeba are the cytoplasm and organelles. Boop, and there it just went. So those are all the organelles leaking out. All the chromosomes just got squirted out, right? The nucleus popped. So all these little bacteria here would have a field day being able to eat all this stuff, except of course, they're all gonna die in the end, right? Everything goes into the disinfectant dish. But it's really interesting that the cover slip actually can fall down and squish a little tiny organism like the amoeba and cause it to pop. It can actually squish things like paramecia too, and they also pop. But all the organelles, all these things are all the organelles inside, and they're all leaking out. So these are organelles, right? Things that we can't, tell what they are, but mitochondria are there, lysosomes are there, all kinds of vesicles are there. So but also take note of all these motile bacteria, right? So we can observe motility in this video as well. Right, a very active one there. Another one up here. So, so something else that we're going to talk a little bit about, since we're talking about protozoa and things, when it comes to termites, we all know what a termite is, right? A little insect. What does it eat? What do termites eat?
Wood.
Wood, right? So they eat wood. And they do damage to your house because they eat the wood in the house. But again, they help to eat, dead trees and things. But remember, wood is made of cellulose. Can animals digest cellulose? Hmm? I mean, can you get nutrition from just eating lettuce?
No.
No, because even though cellulose is made of glucose, like starch, like glycogen, right? There's tremendous potential energy in cellulose. The way the glucoses are bonded to each other, that kind of linkage, animals do not have the right enzyme in order to split those glucose molecules and break them apart. So then how do animals survive? By eating plant matter, right? Because lots of animals eat wood and grass and other plant matter, so it's all cellulose, but they don't have the ability, right, to make cellulase to break it down. So what do? Well, they have to rely on microorganisms, in particular bacteria, but also protozoa, mostly bacteria. So here we're looking at the gut contents from a termite. So a live termite that I squished, and I made a video of the microorganisms in the gut. And we do see some large protozoa like this one down here. But we see these beautiful spirochetes, right? See the spirochetes wiggling around here? So lots of beautiful spirochetes inside the termite gut. But it's these microorganisms that produce cellulase. And so that allows termites to eat wood, and the cellulases break down the cellulose into simpler sugars, and then the termites can survive. But without all these microorganisms, termites could not eat wood. Again, we see these beautiful spirochetes wiggling around, and some other motile bacteria inside. The termite gut is full of bacteria and protozoa, and they help the termite to digest wood. And termites are also very nutritious, so they're good to eat. But all these animals that eat wood and just plant matter, they have symbiotic microorganisms in their guts, which actually break down the cellulose for them. So any questions on protozoa? So if you see an image of an organism with flagella, you should know it's flagella or cilia or pseudopods, right? We obviously, even during face-to-face, we would not have any wet mount slides of protozoa because that's just too hard to deal with for a lab exam. But you may see some images, and you should be able to tell me the types of locomotion present. You should also be able to distinguish trypanosoma from plasmodium and know all about them. So, any questions about the protozoa? No? Well, protozoa is the only thing we're going to do in lab, so if you have no questions, that'll do it. All right, you guys have a good weekend. I'll see you on Monday.
Thank you. You too.
You too. And
Thank you.
Melanie, you stick around. All right, so Melanie, stick around. All right, so bye, everyone.
Hello.
Hey, Melanie. So,
Hi, you
think,
can hear me.
yeah.
Really. I saw your email. I didn't respond because I was like, oh, I'll just tell him after lab. And then right now I just logged out and I'm like, I forgot to tell. So I spoke to my boss and I told him that I'm taking this class and this and that. And I feel like it's not really necessary to switch out. Like I had chosen this time for a reason. I mean, I don't know. I'm very like indecisive about it because at first I'm like, well, I can work, those days. But at the same time, it's like it's not really like why do that all of that,
Mhm.
I think I'm just gonna stick with what
Okay.
this course. But I'm sorry for I was so sure about it and then after I like really thought about it and I spoke to him and he was like, it's okay, like you could take these other shifts or you could do like half day and stuff like that. So I think I'm good as of right now.
Right. Okay.
But thank
Okay.
you very much for looking